All right, let's go ahead and get started. Now that John Luke is here, we're ready to go.
Sorry.
So today what I'm going to do is I'm going to try to open your mind up a little bit to make space for all the stuff you're going to have to cram in for the rest of the semester. Again, the hardest part usually about learning something new is letting go of the stuff you think you know that isn't right. Now, and I'm going to tell you lots of things today about things you've learned in science classes in the past that are not correct. That doesn't mean that your instructors have been lying to you all these years. But you have to understand that anybody is ready to receive information or understanding at a particular level at a different time, and then they have to build that up. It's like peeling an onion, right? Kind of in reverse. And just like physics goes from atoms to electrons and protons and neutrons, and then to subnuclear particles, etc., we have to kind of work our way through levels of sophistication in these biological concepts. Also, there are some concepts that you think about or that are buried in your understanding of biology that are relics of biological concepts from a couple of hundred years ago. And I can't strip those out of your brain. I can't strip them out of mine. But I want you to be aware of them so that when you see them in your thinking or in your words, you recognize them for what they are. So we're going to start with the evolution of evolutionary thought. So even before people knew about biological change, there was a lot of interest in organizing how things in the world were related to each other. And people like Aristotle spent a great deal of effort organizing things, materials, animals and plants, everything, into a linear arrangement. The assumption was that there was a linear organization of all kinds of things. And the axis of this, of course, is superiority to inferiority, right? Things are better than each other, and they're somewhere on this linearity of superior to inferior. And so fish are better than amphibians are better than reptiles are better than birds, etc., etc. And then you can zoom in on that and look at particular kinds of things. And so not only every kind of thing is in an arrangement, but every species, and at least in the case of people, kinds of people, and even individuals, right? This is a very hierarchical view of how things work in the world. No surprise, of course, that whatever kind of person was making this arrangement, their kind of people ended up on top of the pile, right? And not just living things, but other kinds of things, liquids and minerals and glass. This is where the notion of transformation of lead into gold comes from. Because gold was one level above lead, and they figured that they could do something to make lead a little bit better and turn it into gold. Later on, when it was thought that the elements were earth, wind, fire, and ether, the notion was they needed to add a little bit of ether meaning sublime, and turn lead into gold that way. Because in many ways, of course, the properties of lead and gold are similar. Now this runs into some immediate problems when you think about it from today's perspective. You get up here to reptiles. Obviously, birds are better than reptiles, and mammals are better than birds, at least according to this way of thinking. But are birds really before mammals? The problem with this is that you're jamming what isn't a linear relationship into a linear relationship. I'll point out that there is one scientific area in which a relic of this notion still exists. And there's two of them right here on the wall. The periodic table of elements. So in the periodic table, you have a linear arrangement by atomic weight, right? Or atomic number, to be more specific. And then those lines are broken up and stacked onto each other to have elements with similar properties in columns. But it's fundamentally a linear arrangement by atomic number. Because elements are related to each other in a linear fashion, based on the number of protons in the nucleus. But that's not true in biology. And yet, when I go to the RNA meetings, I think they're in Madison, Wisconsin this year, and we'll talk about, there'll be people there talking about splicing and yeast and all this kind of stuff. I guarantee you that I will hear some well-funded senior, research scientist talk about how the system they're interested in is conserved clear from yeast to humans. And they'll do the hand thing, right? Yeast to humans. Yeast to humans. That's what that person is referring to. They don't think that, but that's where it comes from. The notion of higher and lower eukaryotes. That's what that comes from. Higher and lower organisms. Whenever you hear higher and lower, ask yourself, what does that mean? What does it really mean? I'll guarantee you that it's not right. So the first notions of evolutionary thought, and these go way back before Darwin, were the evolutionary ladder. The notion was is that this chain of being moved up. An organism would evolve from one group to the next as they got better and better. So here's number one, right? Homo sapiens. This isn't true either. Humans did not evolve from apes. Humans are apes. It's as simple as that. We didn't evolve from chimpanzees. Chimpanzees and humans have a common ancestor, and that common ancestor was neither a chimpanzee nor a human. Missing link. I told you about higher and lower eukaryotes. Missing link is another one. What is a missing link? A missing link is something in this chain of being where someone thinks there ought to be an organism and they haven't found it yet. It's the chain of being, a missing link. Presumably between the evolution of one kind of organism to the next. It's the half step. There's no such thing as missing links. Well, of course, Charles Darwin thought otherwise, and this is one of my favorite figures. There's a ton of information in this image. At the time that The Origin of Species was published, including an image in a paper or in a book was a really, really big deal, right? They had movable lead type by then, and they could arrange the type to spell out the words and everything. But graphics had to be carved out of a piece of wood or lead and included in the typesetting machinery. It was inordinately expensive. And in the book on the origin of species, there is only one figure. There's only one concept that Charles Darwin thought was so important and so graphically centered that it had to be presented as a graphic. And this is it right here. And so here we have time slices, observations at various time points through some history. And we have a number of species here along the bottom, A, B, C, D, E, F, etc. And then what happens to them over time. And you'll notice some really interesting things that evolutionary biologists today still argue about. You'll notice that most of those species undergo little or no change whatsoever. They continue more or less as they are. They're perfectly well adapted to their current environment. And there's no need for any particular evolutionary change, and they continue as they are. This is the notion of punctuated equilibrium. That, again, it's a really interesting area of evolutionary biology these days. Most of those go extinct. Some of them persist to the final time slice here. You have two groups, two species, that undergo a lot of evolutionary change. Presumably, they're not particularly well adapted to their environment, or their environment is in flux, and so they're constantly having to change to evolve to match that environment. They're in disequilibrium somehow. And they throw off lots and lots of species, most of which go immediately extinct, but some of which survive. And maybe these are subspecies, maybe they're legitimate species, you don't know at this case. Most of these go extinct, and at the final time point, this single species A is now represented by 3 species. And this single species I by 2. Organisms evolve not in a linear progression, not in a direction, but by adaptation to their local environment and by diversification. They change over time, but not in any particular direction, just whatever is needed at those moments, every evolutionary time point. What works a little bit better or a little bit worse, that's how they evolve. There's no advancement. Well, that tree is an abstraction. Yeah. There's no the right and left axis is irrelevant. It's just in that particular case, the notion is that the further apart they were, the more distinct they were in some measure. So that picture was an abstraction. Ernst Haeckel really liked The Origin of Species and the related books of Charles Darwin's. And he's a very famous artist, scientist at the time. We'll show some pictures of some unicellular plankton later in the semester that are his drawings. Really amazing. He did a bunch of embryology as well, which was a little not quite so good. But he took this notion of an evolutionary tree and put real organisms into it. And that's what this is. And this is really actually pretty good. And so here's monarans. These represent basically bacteria. These are various kinds of what people call protists, flagellates, myxocytes, etc. And they're divided generally into plants, protists, and animals. We would disagree with that today in a variety of little ways, but the notion is about right. It's correct in some really good ways. So, for example, you'll notice that monarans, bacteria, aren't shown down here as the ancestors of modern things. They're shown as modern organisms, right? They're alive today. There are a bunch of algae that are shown here as related to plants. That's at least partially true. Animals are not seen as descendants of various kinds of protists. This is all true. And of course, these were sorted out based on morphological characteristics, which in the case of eukaryotes, which obviously this focuses very heavily on, can sometimes be a pretty good guide to how organisms are related. Now, I would argue that amongst these organisms, it's not so good, certainly not amongst the fungi, wherever they are. I don't see them at all. But in general, this isn't a bad first try at it. How many of you grew up in high school biology looking at this thing? The five-kingdom tree, right? Bacteria, protists, plants, fungi, and animals. This is a step backwards. This is worse than Ernst Haeckel's tree. And it's worse for a variety of reasons. Number one, bacteria are shown to be the direct ancestors of eukaryotes. This is false. Most of these trees don't show particular phylogenetic lineages. They just show the groups. They don't show the branches. So they're trying here to show some branches. But clearly, something is represented by this vertical axis, right? What is that?
Higher, like
superior, right? Superior, better. Vascular plants are better than bryophytes. Mushrooms are better than yeast. And humans are better than anything else. That's what this means. There's no timeline here because these organisms are alive today, right? Now people will wave their hands and say, well, that's not really what it is. It's not superiority, it's complexity. And yet you'll notice that none of these branches here in the animals that go to things like tapeworms that are highly reduced and highly simplified, those arrows don't turn around and go down again. This is not complexity. It certainly isn't time of emergence. This is one thing that I hear a lot of time. Well, this is when the groups first evolved. the timeline. So animals didn't evolve until such and such a time. So that's why they're up there. E. coli and Salmonella separated 100 million years ago. How come they're not up here? And the obvious reason is because this isn't about time. This is about superiority. And I have to argue that scientifically, I don't know what superiority means. That's not something I can measure. So how do we do better than this? How do we actually measure evolutionary relatedness? Put numbers on it so that we can construct an evolutionary tree that is objective and quantitative. The way that we do this is with gene sequences. Now you can do this with morphological traits and that kind of thing, but of course you're limited to organisms that share those morphological traits. I mean, you can't measure facial differences in bacteria because bacteria don't have faces. So you have to use something more interesting. And one thing that all organisms share is genes. And so you can study genetic similarity and differences. And when you do that, you get a tree that looks something like this. Now this is an unrooted tree. It turns out that when you create a quantitative evolutionary tree, in order to have a root to the tree, in order to know where the base of the tree is, you have to have an outgroup. You have to have something to compare it to. And in a universal tree, you can't have an outgroup, right? That's what universal means. I'll tell you later on that there's actually a trick around this. But for now, let's just go with the evolutionary tree as it is. It turns out the root is about here. And we do have three major kinds of organisms, but they are not plants, animals, and protists, as Ernst Haeckel suggested. They are bacteria, eukarya, and archaea. Bacteria we all know about. Archaea, you've heard about archaea, and you think they're really weird and that sort of thing, and that's fine. We'll talk quite a bit about archaea in this class. Archaea are not kinds of bacteria as they originally were thought. And then you have eukarya. This requires a little bit of explanation. What are eukarya? Most eukaryotes are amalgams of bacteria and eukarya. And so if you look at a human cell, it's got a nucleus, it's got cytoplasm, and it's got mitochondria. Those mitochondria are not eukarya. Everything else is a eukarya. It has that common ancestry. But the mitochondria are permanent, obligate endosymbiotic bacteria. Millions of years ago, probably billions of years ago, an ancestral eukaryote formed a symbiosis with a an alpha proteobacterium, and that relationship became permanent for both host and symbiont. And those organisms persist today in most of the eukaryotes we know about, not all of them, or maybe not all of them, and in both the mitochondria and eukarya. But the eukarya here, you're talking about the nuclear cytoplasmic lineage. In phototrophic eukaryotes, there's a second endosymbiont that is the chloroplast, or plastid of various kinds. And whether those, well, it's absolutely clear that a variety of eukaryotes took up plastids independently. Exactly the relationships there are so far still uncertain in many ways. And so eukaryotes are amalgam organism. When I talk about eukarya, I'm talking about just the nuclear cytoplasmic lineage. And so these are represented by genes in the nucleus and genes that originated in the nucleus. Now there's no up or down in this. There's no superior, inferior. There's no more or less complex. The measurement here is evolutionary distance, just how different are the sequences from each other, and they're measured along the branch length. And so I can compare, for example, and show that Synechococcus, it's an alga, and Bacillus are about as similar to each other as yeast and humans. You find that surprising? Here's another one. You You guys know what dictyostelium is, a slime mold? Here's humans and slime mold, about as similar as Chloroflexus and Bacillus, two bacteria. And so these represent divergences in molecular sequences in the genomes of the organism. This tree is objective. It doesn't matter who makes it. It comes out the same. And it's quantitative. You can take measurements across this tree and understand something from those measurements. You can also argue about the details of it, and that's fine. So when this first came out, this kind of a tree first came out in 1977. I took general microbiology in 1977. It made the cover of the New York Times. The discovery of a new kind of organism that nobody knew about. They were the methanogens down here, the archaea. The instructor for that class completely botched the description of it. As far as we students knew, we were talking about silicon life forms or something like that. They're not silicon life forms. They have the same DNA, same genes, same membranes, all that kind of stuff. But they are not related to bacteria as they had previously thought to be. It turns out that many of these organisms have been studied for a long, long time. Sulfolobus, for example, was thought to be some weird member of the Pseudomonads group. Methanobacterium, I don't remember where those were. Haloferax was another Pseudomonad. And nobody realized that they weren't just bacteria, that they were something really, really different. When you look inside at how they work, they're really a lot more similar in many ways to eukaryotes than they are to bacteria. And that's because they share a common ancestry with eukarya. If you're going to divide this tree in two, it would be bacteria and everything else, not eukaryotes and everything else. I'll get to that in just a moment. This kind of tree also, by the way, proved that endosymbiont theory I just talked about, because you can analyze the genes of eukaryotes separately. You can take mitochondria have DNA, and you can pull that DNA out, sequence it, and include it in this analysis. And sure enough, they show up here amongst the proteobacteria. Likewise, plastids show up here amongst the cyanobacteria. There's been some thought that the spindle apparatus and flagella, the whole microtubule structures in eukaryotes, might also be the result of one of these symbioses. The data for that is poor, and since there's no DNA involved, if they ever had it, they lost it, then it's not something that can be addressed by this method. Yeah?
Do you have any theories about what sort of what modern bacteria today might have been similar to the one that became endosymbiotic as mitochondria?
Yes. So if you look at so the question is, are there any organisms alive today that might resemble in some way the progenitors of the mitochondria? It turns out that when you do the analysis carefully, the mitochondria turn out to be very closely related to, and it's difficult because of differences in evolutionary rate. They turn out to be members of the same group of alpha proteobacteria that include things like rickettsia, the spotted fever disease-causing organisms, and a bunch of other organisms that are all symbionts of eukaryotes. Some pathogenic, some mutualistic, but they're eukaryotic symbionts. And the mitochondria have clearly taken this one step further. Likewise, the plastids, by the way. They're related to prochloron, which is one of the green organisms that live in association with various creatures in coral reefs and turn them colors. Yeah? A plastid is a generic term for chloroplast. Chloroplasts, formally speaking, I think are generally are what people call the plastids of plants, of green plants and green algae. But things like diatoms also have photosynthetic organelles, and they're not usually called plastid. They're not usually called chloroplast. Plastid is a more general term. There's some organisms that have those organelles, and they aren't even photosynthetic anymore. There's some parasitic plants. The Spanish moss has plastids, but they're not photosynthetic, and so they're still called plastids. All right.